Welcome back to our final video on Exam 1 topics for MMP. In this video, we'll go over fat absorption, cholesterol synthesis, protein metabolism, and the urea cycle. So let's begin with fat absorption. So triglycerols, which are a fat storage molecule, enter our body through our diets. What will happen next is when they're in our small intestine, the gallbladder will secrete what are called bile salts. And these bile salts will surround our triglycerides as they pass through the small intestine. We have then an enzymatic process of cholipase and lipase, which will break down our triglycerides into fatty acids and a 2-monoacylglycerol. Now the way these get signaled is by the signaling hormone CCK, which is detected when food enters the small intestine from the stomach. That acts on the liver, it acts on the gallbladder, and it also acts on the pancreas. And you get secretion of enzymes as well as bile salts, such as the lipase enzymes we just talked about. Now what happens after these are degraded by these enzymes is they actually are resurrounded by bile salts once again. Just this form, they're the products instead of the triglycerides, they're the fatty acids and the 2-monoacylglycerols. And these will essentially bind these kind of like pockets with the lumen of the small intestine, so our products can get essentially absorbed by the cells of the small intestine. And something else that also happens is a little bit of our food and fat gets lost with the bile salts. About 95% of our bile salts actually get reabsorbed into the liver from the cell walls. This also happens with drugs as well. This is called enterohepatic circulation, in that products will go to the liver and get metabolized and then be continuously resecreted. In drugs, for example, this can lead to more drug degradation. And for bile salts, it allows them to be reused multiple times and not be broken out and expelled by the body. So what happens next? The triglycerols are actually reformed from the fatty acids and the 2-monoacylglycerol, which I know seems a little bit redundant in the fact that we just took all that time to break those down. These are reformed in the intestinal cells, and they're conjugated with this protein called ApoB48. This is in the small intestinal cells. There's also another ApoB, it's ApoB100. This is what's conjugated with in the liver. This essentially binds the fats to create something called a micelle. A micelle is similar to, think of it as something almost like a mini cell. It has a like a lipid bilayer, or maybe it's a lipid monolayer, but the point is the hydrophobic ends on the outside and the hydrophilic ends on the inside with all the fats, so it's able to travel through blood and kind of store our molecules of fat, of our triglycerides. Something else to note about ApoB48 and ApoB100 is they actually come from the same gene. They're just cut differently in gene like transcription modification. That'll probably be a question. I think that was a question for us. So know that those are from the same gene. They're just modified a little bit differently. But with our micelles, then, while they're traveling through the blood, eventually we'll come across to enzymes called lipoprotein lipase, which will bind onto these triglycerols and separate them into glycerol and then fatty acids. And these are able to actually pass across the membrane into the cells wherever they need to be in the body. So we're no longer in the small intestine here. We might just be in some random portion of the body, probably near adipocytes, because that's the main fat storage molecules. And then when these micelles are broken down by this lipoprotein lipase, and so are the triglycerides, something that actually remains is the ApoBs won't, but the ApoE, which is just another protein that's expressed in these, will remain, and that'll go back to the liver. Now, a drug that may work on this system is called orlistat. You'll learn about this in OTC this semester if you haven't already. This essentially, it's a weight loss drug, and the way it works is it prevents fat absorption by inhibiting this first step of the lipase in the small intestine. So our triglycerides never get broken down. They're never able to absorb. So this is essentially how they make you lose weight, in the sense that if you have a high-fat diet, you're unable to absorb your fats. However, this isn't the best drug. It's actually researchers at UC doing research right now implying that this may have certain carcinogenic potential, as well as the fact this drug has some pretty nasty side effects. So usually we don't recommend this in an OTC setting. We try and go for diet and exercise, but it is a product you may encounter, so it's good to learn about. So now let's talk about how fatty acids are synthesized for the body. This process will take place in the liver. Essentially what this will do is in the mitochondria, it will convert pyruvate to acetyl-CoA using pyruvate dehydrogenase. You may remember this enzyme from earlier steps, I think all the way back in the first lecture. And eventually this will enter the TCA cycle to become citrate. Where this deviates a little bit though, is the citrate will actually work a little bit. The TCA cycle doesn't have one direction, but how it's traditionally drawn is oxaloacetate turns into citrate. In this case, we do the opposite. And the reason that it's usually considered that way is because this actually takes an ATP in order to do. So this is a process that will occur mainly when energy is high in the cells, which makes sense, because if our energy is high, then we don't need a ton of energy right now, and that would make more sense to store with fatty acid. Something else to mention is that insulin actually upregulates pyruvate dehydrogenase. Another thing that kind of makes sense logically, as more things enter the cell because of insulin, we'll have more energy, so we are like able to do this process more, is the long and short of it. The enzyme that is the rate limiting step for this as well is acetyl-CoA carboxylase in fatty acid synthesis. And the reason that this happens is because it requires biotin as well as CO2 in order to add carbons on. And something else to mention with this reaction, as we're adding on fats, it goes through this pathway. This isn't the most important, well, this is the TCA cycle. This is the pathway where malonyl-CoA is added on, and essentially this adds on 2 carbons at a time for fatty acid buildup. The enzymes aren't the most important except for acetyl-CoA carboxylase, since that is a rate-limiting one. See that right here? Converts acetyl-CoA to malonyl-CoA. And this is upregulated again by insulin, and this will sort of make it go forward. This is the enzyme complex over here. It goes 2 carbons at a time, so again, kind of the antithesis to beta oxidation. And something else to note is, I'd say the last important thing about this is that it does require an NADPH, which, if you may remember from one of our older videos, that comes from the pentose phosphate pathway as the primary producer of those. I'd say those are the major takeaways from this. This wasn't something that was tested on super hard on the exams, but I would say know acetyl-CoA carboxylase, know that it requires ATP and NADPH, and know that also to move forward, there is a requirement of biotin and CO2. So something else to note is that malonyl-CoA actually downregulates CPT1. This is a protein expressed in mitochondria that allows fatty acyl-CoA to pass through. You may remember that from our first, I believe it was the first video in this series. Or no, it would have been the second because it was beta oxidation. Since this process is opposite to beta oxidation, the breaking down of fatty acids, it would make sense that a substrate of the fatty acid synthesis would actually inhibit this, because if we are trying to build fatty acids, we don't necessarily want them going into the mitochondria to be immediately broken down. So that's why logically it kind of makes sense that this is an inhibitor. So yeah, just know that if things are being synthesized, they're going into the mitochondria less. Something else to note is the process we've talked about so far makes saturated fatty acids or fatty acids without double bonds. If we do want double bonds for an unsaturated fatty acid, we require 2 things. We require, it's not on this diagram, but oxygen and NADH+. Which essentially means this is a process that requires energy, and this occurs in the cytoplasm of the cell. This is also regulated by a cytochrome, which, if you remember, is just essentially an enzyme with iron, specifically cytochrome B5. I don't think you'll get asked that, but there's always a chance. I would mainly know that it's a cytochrome. And then what can we do with fatty acids now that we have them? So one thing they can become is these eicosanoids. I know they're a little bit small in this cell. But essentially, what these are is they are inflammatory mediators. They can be prostaglandins, leukotrienes, and all you really need to know is that these cause inflammation, they can cause pain, they can cause swelling, which all sound like bad things. Of course, like in regulation, right, if the body is actually in pain, these are very useful. But these are actually moderated by an enzyme, cyclooxygenase 1, which is actually what aspirin is a irreversible inhibitor of. So the reason that we feel less pain when we take aspirin is because it's inhibiting this class of molecules for making prostaglandins, essentially reducing the pain we'd feel from some form of inflammation, whether that be necessary or unnecessary in the body at that time. Something else is insulin will actually upregulate lipoprotein lipase. So as things are going into our body, or as insulin is promoting things to go into our body more, we're actually changing gene expression to increase this lipoprotein lipase protein so that we essentially encounter these micelles more with triglycerides. They just get bumped into more often because there's more of them statistically, meaning we can bring up more triglycerides and fatty acids into our cells. Well, they won't be triglycerides when we bring up. They'll be glycerol and fatty acids. But essentially, yeah, so as we actually get more insulin in our body more regularly, even our genetic protein expression changes. So that's just something to remember. And then also, we can increase these fatty acids being released from the cell through glucagon, although these don't really cause a change in protein expression. Okay, so now let's talk about cholesterol synthesis. It begins with many things, like with acetyl-CoA, and essentially it will do this through the enzyme HMG-CoA synthase, which will create this intermediate right here. That's not incredibly important because the next step is much more interesting, this HMG-CoA reductase. That may sound familiar to some of you. This is the primary mechanism how statins work, which are cholesterol controlling drugs. They're HMG-CoA reductase inhibitors. So by inhibiting cholesterol, you actually start by inhibiting it at the very beginning versus the very end, and only the second step. And this will make this product mevalonate. Now, the next few steps, you don't really need to know what they do specifically. The only thing I would recommend knowing is that all three of these require ATP input. So cholesterol synthesis is a very high energy process. Oops. There we go, my pen wasn't working. High energy. Now there's going to be a series of carbons added. These are through high energy phosphates. Something else about these is some of these further steps will begin requiring NADPH. Again, that's from our pentose phosphate and also from building fatty acids, so another use for that. But really the only next major molecule you need to know is squalene. And the reason squalene is important is the carbon backbone has 30 carbons, and squalene is the last time squalene has 30 carbons as well as cholesterol. So this is the step where carbon is finally done being added. There's then a whole complex of enzymes, which eventually, they're called cyclases. They'll go through a bunch of steps to make them cyclized rings, your four-membered rings that are common of cholesterol, until you finally get cholesterol. The rate-limiting step of this whole thing is HMG-CoA reductase still. So really important enzyme for this process. Not only the fact that it's used in statins, but also just for this process. It's actually regulated in the body by three things in transcription. So we can genetically express less of this or make less proteins based on this. Proteolysis, which is essentially degradation by acidic conditions, as well as inactivation by phosphorylation. So if we take a molecule that has some kind of phosphate on it, some energy carrier, we can actually switch this off until we need it. So it's pretty handy that we have all these ways to regulate it, because cholesterol isn't always needed in the body. So dietary cholesterol in high amounts can lead to conditions later down the line, and this is kind of how our body combats that by only making sure it's on when it's needed. So now that we have our cholesterols, how do we get them where they need to go? Essentially, once we have the basic molecule of cholesterol, they're converted to three main things. You have these cholesterol esters. This is an ester group, if you remember from OChem. This group with the carbon group as well as two oxygens attached to it. Bile salts, which you may remember from fat absorption, as well as biliary cholesterol. The way bile salts are created is there's a whole string of processes that cycle it through, but the main thing is, in order to make this, you use NADPH. Again, this molecule is coming back from the pentose phosphate pathway. And this system is also a cytochrome system, so enzymes using iron will work up here in order to kind of start this process and get the bile salts to where they need to be. And then our main proteins, you may remember that fats can't be transported easily in the blood. So we have our apoproteins as well as kind of a lipid layer to create our micelles. The ones that will carry cholesterol are ApoB48, ApoB100, and ApoE. Something else is that when bile salts are secreted, again, we mentioned this in fat absorption, most of them will get reabsorbed into the liver, and that can be reconverted to cholesterol, that can be reused as bile salts, but we like conserving these if we can. Okay, so now how does cholesterol actually get into the cells? As I mentioned before, the micelles are conjugated with ApoB100 and ApoE. What we can do is that LDL receptor, or low-density lipoprotein receptors, actually can mark these for uptakes, and they go into the cell through endocytosis. Essentially, then these receptors are recycled, they go back to the surface, and the endosome or micelle essentially breaks down into the cholesterol we have. Now, what does cholesterol do in cells? And the main thing cholesterol can do is to make hormones, steroid hormones specifically. The very first one from cholesterol is pregnenolone. It then can vary into multiple different things. The first familiar one may be progesterone, but it can also be things like cortisol, which is a stress hormone, aldosterone, which regulates blood pressure. We have testosterone, estradiols, so a lot of the sex hormones as well. None of that pathway is incredibly important, but just good to note that that's how we get cholesterol into our cells, and those are some of the hormones that can be made. Vitamin D is also synthesized with cholesterol. You may or may not know that we can both get that from dietary means and also from we can use cholesterol as well as energy from the sun to kind of catalyze this process in order to make intracellular vitamin D. And this is important because this kind of regulates calcium and phosphorus in the bones. So that's why vitamin D is pretty useful to have on hand. Okay, now let's talk about the degradation of proteins in the diet. So it begins in the stomach digestion with zymogens, which are essentially precursors to enzymes, which are activated by different things. The main activator is actually trypsin, which is a protein itself. And the way this gets activated is by the detection of peptides in the stomach. And you can see that all three of these other enzymes are actually activated by trypsin, with the exception of pepsinogen, which is activated by the acidic conditions in the stomach. As we get more food in the stomach, that acidity rises, so that'll increase. And that can break down peptide bonds in proteins. No one zymogen can break down everything, so that's why we have a series of these in our body to deal with different kinds of amino acids. How are amino acids absorbed then? Well, like the breakdown actually occurs in the stomach. Absorption occurs in small intestinal cells and large intestinal cells, but primarily small. You may remember the amino acid sodium transporter that we also had with glucose. It's extremely similar here in the fact that this is a passive form of diffusion since we're moving sodium with its gradients, even though amino acids might not necessarily be with their gradients. And the way we do this again is with that potassium, sodium, ATPase enzyme. So now that we have amino acids in the body, what can we actually do with them? One of the more important amino acids is glutamate. This can actually be turned into alpha-ketoglutarate, which, as you may remember, is part of the TCA cycle and also transporter systems in the body. And this actually creates an NADPH. So that's another way, besides the pentose 5 pathway, that we can create that. Again, these shuttling mechanisms, this goes back to the electron transport chain, being able to convert things like pyruvate into oxaloacetate and transfer in and out of cells can work by having these intermediates like glutamate. So having those nitrogen processes is really important. Something else glutamate can do is it can be converted into glutamine through a series of processes, which can be used to excrete urea in the liver. Alternatively, it can also become alanine. Or this isn't exclusive to glutamate, but amino acids in general can donate these nitrogens and convert this process to alanine. And if you remember, alanine was the primary amino acid used in gluconeogenesis in order to create new glucose for the body. So these are also a viable energy source. So they're just a few of the things we can actually use them for. And then finally, excreting nitrogen from the body. Nitrogen is supposed to be a balance in the body. It's obviously very useful in things like transporter and energy creation, but too much of it can lead to toxic waste products, such as ammonia. So we have systems in place for kind of finding a way to detoxify this ammonia and also excrete it from the body. So the first kind of step to that is our carbamyl phosphate transferase, or our CPS1. We take inside the mitochondria an amino acid and combine it with carbon compounds eventually in order to get carbamyl phosphate. This actually takes 2 ATP, so it's very high energy input. We then can convert this into citrulline, which will exit our cell and go through arginosuccinate synthase, essentially following this pathway fumarate. You may remember that from TCA cycle. That can also branch off right here. This is the waste product of this cycle. So this actually creates more energy. So even though we did input some energy with ATP at the beginning, this can at least be cycled back into the TCA cycle to hopefully generate some more into arginine. And eventually arginase will convert this into urea, which is excreted in urine. This over here is called the Krebs bicycle. You can see that essentially here is Krebs, but with the addition of a nitrogen from glutamate, we can actually turn this into aspartate, which meets up with the urea cycle. So these kind of have an overlap. Another overlap we just pointed out was the fumarate, which starts up there. So this can kind of recycle these carbon compounds. They can be amino compounds, they can be just hydrocarbons. It really depends on what we need them for at the time. But yeah, and then beyond that, arginine and N-acetylglutamate, those both uprate the CPS1 protein. That's probably the main protein enzyme you're going to want to know for the urea cycle. It's your primary one. It's the one that takes the ATP at the very beginning. It's the first step. These both upregulate it, which make a lot of sense. If we have a lot of compounds with nitrogen, then we're going to want to excrete a lot of urea, so we don't have too much. And with that, good luck on your exam, and I'll see you in the next one.